Well, according to the other participant here, I think I'm a little bit the odd man out because I'm not directly involved in development of EVs or in research, but we're coming from a supporter side. So Anasystem is a distributor for particle analyzing instruments based in Germany, and we are offering solutions for particle analysis. And one solution is a system from the US company Spectrodyne. It's called the NCS1, and this is a single particle counter, generally based on the Oculta counter principle, and it can be seen as an orthogonal method to established methods like NTA. And yeah, this is why I'm here today, to present the system to interested people and maybe to spread a little bit this technology and the big benefits for EV research.
To be successful in the EV market, you have to identify the challenges in the field and the people that need those challenges or need those challenges solved. And so you need to make sure you're addressing a pain for the customer to make sure that they're able to answer the hard scientific questions that they're posing. So as an example, I think people are really trying to understand EV heterogeneity and be able to look at single vesicle analysis. So being able to look at single vesicles to really understand the population dynamics and how those biomarkers can be expressed in either groups or on single vesicles or co-localized. So that's an example of addressing a pain in the market.
So the success of NanoView, I think, is kind of stems from 2 points. Is the multidisciplinary team we put together. Mostly our team consists of engineers, software engineers, and cell biologists. So we get to attack the problem from different viewpoints. But also what really excites us about NanoView, about extracellular vesicles and EVs is it's a rapidly growing research field, and people were very willing to collaborate with us to better understand the problems the field is facing and kind of work with us hand in hand to kind of make sure we're on the right track and solving the right problems.
So if you want to keep a good relationship with the investors, the best way is to be open, to be transparent, and it is important to involve the investors in your daily business. And you need to make them comfortable that the investment that they did, it's really working well. And because you need to be transparent with them, it is really important that you can share almost everything about the business. Don't be scared to share those things because, you know, the investors, they are there to help you, so you can say anything. And be comfortable that they are there to help you and not just to, don't be afraid of sharing important matters.
INDEX is a project devoted to develop an analytical platform for exosome collection from biological samples and analysis. So the idea is that if we want to use exosomes in diagnostics, we must be able to recover them from complex biological fluids with high specificity and characterize them on a platform that is easy to use, where people without particular skill can use it in a context of a clinical application. And this is the type of needs that are behind the INDEX project. And why in Europe? In Europe because we don't have in Italy the expertise that is required to do this. So we have collaboration with partners from other countries, from Paris, from Denmark, and also a particular characteristics of this project is that we have a partner from United States. So we were able to convince the European community that we needed technology that was developed in Boston University, and that now this technology is now incorporated into our platform. So what could be the future interest of INDEX is that if we succeed in developing this prototype, which now is very manual and very, let's say, not completely defined, we will be able to advance the field of the use of exosomes in diagnostics, and we will probably apply for another project where we will be more focused for industrial exploitation of the technology, where we really will produce this platform, and with this will induce, of course, the know-how of the European community into this kind of field.
We are here to present 5 ETOPEN projects and the clustering of the 5 ETOPEN projects as and the significance to have 5 ETOPEN funded by and currently running in the context of this funding scheme of the European Commission, and these highlight the importance of a main theme that is the common and a common issue among the 5 of us, that are the extracellular vesicles and their use thereof for breakthrough technologies indeed. So I think that what we propose, so what the projects propose, would be of high impact for the European society, either for health or for cosmetics, for instance, as a possible field of application. And I'm also would like to highlight the importance of having this meeting and this clustering and bringing top scientists in the field here in Palermo. And I think that this is going to be also very impact for our community, since coming from a small island on the south of Italy and bringing these very important issues on Europe would be of very high impact indeed.
Okay, so the EV Foundry project is based on the idea that we need to produce high amounts of high-grade extracellular vesicles for next uses in large-scale testing and analysis, and so we need it to, let's say, to fill the gap that we have between the current state of the art of science and technology and the next level needed for real translation into clinics and other technologies of extracellular vesicles. So this is a kind of contribution that can help at different levels, at the regional and European levels, because this is a technology that can keep the level and the pace of Europe to be updated with what is going on in the rest of the world. And so the meaning is really at the international level.
Okay, so Gladiator actually is a very fundamentally new approach to science, in a way that we integrate ICT, biology, and really to a loop of regulated sort of therapy processes that impact the brain. So basically then it brings total novelty on the radio frequency studies, in a way that we actually can integrate radio frequencies to a sort of a setting where we can kind of have an impact on medical kind of therapy opportunities into complexities like brain, where the exosomes actually are the responsive players, that they go across the blood-brain barrier to actually deliver this therapeutic property and reprogramming property to the brain gliomas, to actually then set up a loop that is integrated also to a readout of this recovery process, so that the ICT bio integration happens. And the kind of unifying scientific theme is a new emerging field called molecular communication, and this actually is one of the scientific sort of rationale behind the whole Gladiator innovation set.
MindGap has started only in April, so we don't have currently a specific impact on what it will bring out to the population. But we expect it to have a huge impact in the future, because MindGap consists in establishing a connection between the mind and the body interactions controlled by meditation processes, and to understand what is the role of exosomes in the middle. So if we can understand what is happening in the crosstalk about the exosomes that are being sent by the brain to the body, we are going to reveal how the information driven through the fluids are going to contribute to health and disease. And to prove that, we have patients from a cancer hospital that are going to be subject to meditation processes and that are going to be tracked within time to understand what is the role of exosomes in this field. So we are expecting to have a huge impact, a global impact in health of the population in general.
In Vespuras, we are developing a new bioprocessing approach to produce and modify EVs. We have a raw material which is completely natural and renewable and useful in the industrial environment to upscale the EV's production. At this stage, we are in a technology readiness level low. We are exploring the technology and validating the different parts of this technology at the lab scale. I hope that we could make a great impact both in research agenda and in the market with these technologies by 2023 or even a bit more.
The technologies that we are using are not very close to the market because we need to develop collectors and also technology to analyze on chip, and I think the technology is not there. As we have seen, you have two big tables, and also of course we need the data, the experimental data and the clinical data. And then, so I would say maybe 3 years from now, if you ask me, I have a different answer.
I would say that this is very multidisciplinary approach, and then I would say that in overall it takes a long time. I would say at least 5 to 10 years when the overall technology is on the market, but I can see that partly it can be commercialized in much shorter time frame.